Good morning. So for this lecture, we're going to talk about the basic principles of immunologic and serologic reactions. Right. So serology is the study of serum. What is in serum that we would like to study is the antibodies. Right. For purpose of diagnosis, we identify these antibodies because they usually are present in response to an infection. Right. Aside from that, the antibodies are also present in the presence of foreign antigens, aside from microorganisms, like in case of mismatched blood transfusion, so there will be production of antibodies. And also another concern is the antibodies that act against our own cells or our own tissues. So those are found in autoimmune diseases. Right, so we already know this, that the immune system consists of structural cells, organs that allow the organism to recognize and respond to foreign stimulus. Right, so there are two responses, primary and secondary immune response. The first time that the microorganism steps in, it gets recognized by the immune system as something foreign. And the second time it is seen within the system, it elicits secondary immune response. This secondary immune response can be cellular immune response or humoral immune response. Right, so this consists of events that occur during the second or subsequent time of appearance of the same antigen. Right, so we already know what is serum, what is plasma. Serum, both of them are liquid portion of the blood, except that serum is obtained from clotted blood, while plasma is obtained from anticoagulated blood or whole blood. Right. So the next term is specificity. Let us couple it with sensitivity. So specificity is when an antigen meets its corresponding antibody. It's like they know each other, so they are made for each other. So this is a special affinity between the antigen and the antibody. Sensitivity, on the other hand, is the ability of the test to detect the whatever we want to detect, whether it be antigen or antibody, that even if it is present in very minute or small amount, we can still detect its presence. So that is sensitivity. Right, so susceptibility means having little resistance to disease-causing agent. So that means a person is susceptible if he gets easily sick. Right, so applications of serologic test. When do we do serologic testing? One is if we think that there is an infection, so there is a suspected infection, so the doctor may require the patient to undergo serologic testing. Also in non-infectious diseases like rheumatic diseases, right? So, and also to check one's blood type. So even blood typing is considered a serologic test. If you want to donate blood, that is also an indication for serologic testing, because we want to know if the blood that you would be donating is safe for transfusion. Serology helps to diagnose patients with immune deficiencies. Okay, when we say immune deficiency, that means the person is lacking the ability to protect himself with his immune system. For example, there is lack of antibodies, right? So a gamma globulinemia. Right, so for serological testing, the key thing is antigen and antibody reaction. So we know that antigens produce antibodies that are specific, and when they meet each other, they produce a complex. The ability of the test to detect and measure the reaction between antigen and antibody becomes the basis for serology. There are three levels of testing, three tiers. So primary, secondary, and tertiary. Generally, primary binding tests are the one with the highest specificity and sensitivity. Mid-range tests are the secondary binding tests, and tertiary binding tests are classified as more or less for research purposes or in vivo testing. Right, so let's have the different examples of primary binding test. Right, so maybe you have heard of ELISA, enzyme-linked immunosorbent assay, radioimmunoassay or RIA, Western blot, Northern blot, Southern blot. Right, so there is no Eastern blot. Fluorescence test. So those are examples of primary binding tests. Again, they are highly sensitive and highly specific. They are used for the detection of bacterial antigen, viral antigens, fungal antigens, parasites. So it can also be for the detection of antibodies, not just antigens. So again, they are sensitive, they are reproducible. That means they are precise. Right, so let's have one immunofluorescence test. Right, so there are two under this category, FAT, fluorescence antibody tests, direct and indirect. So more or less you have an idea what we are trying to test for, antibody. Right, so direct fluorescent antibody test or direct FAT, FAT. We use these to identify unknown antigens coming from, the various microorganisms. Why do we call it direct? It's because the fluorescent dye is attached or labeled directly to the antibody. So there is a term that we introduce here which is labeling. To label is to attach a substance onto the antigen or to the antibody that makes it easier for the laboratorians to detect their presence. An example of a label is a fluorescent dye. Example of that would be fluorescein. Right, so here it is. The fluorochrome, okay, that is the label. Okay, is fluorescein isothiocyanate, and it gives a yellow-green fluorescence. Okay, a fluorescent substance is one that absorbs light of low wavelength and emits it back at a higher wavelength. So that means usually the wavelength that we use is UV, ultraviolet. And what do we know about UV is that it is invisible. We cannot see UV. However, so it says here that it emits that light, the wavelength, from lower wavelength to higher wavelength. So from 300, it becomes higher, and because it's higher, it becomes visible. Because higher than 300 is already visible spectrum. For the procedure, we're going to have a tissue or smear containing the organism, and we're going to fix that onto a glass slide. Incubate with the antibody containing the label, fluorescent label, and after incubation, we're going to wash to remove the unbound antibody. After which, we can examine under the fluorescence microscope, and the positive result would be something like a glowing in the dark object. Okay, so application. This direct FAT can be used to identify bacteria when the numbers are very low. So that is one advantage of serologic testing. We can We can identify organisms even in low numbers because it's very sensitive. It may also be used to detect viruses that are growing in tissues or tissue culture from infected animals. Okay, for example, in the brain of infected animals in case of rabies, or antigens of HIV on the surface of infected cells like the T cells. Okay, for the indirect fluorescence antibody tests, or IFAT, the unlabeled antibody, take note, unlabeled, is allowed to combine with the antigen, forming a complex. And then the next step is to add another antibody, second antibody, that is specific to the first antibody. And this second antibody is labeled with fluorescent substance. Right, and then the use of this indirect FAT is to detect and identify unknown antigen in this specimen, and also to detect antibodies in the patient's serum using a known antigen. So you can see very readily the difference between direct and indirect FAT. Right, so this is the procedure. A slight preparation of the specimen is made, and we add the unlabeled antibody. Right, and then after some time for incubation, we're going to wash the antigen antibody mixture so that we can remove the unbound component. Right, so the next step is to add another antibody. This is the second antibody that is labeled with anti-species globulin. So we can say this is anti-human globulin. And the excess is washed again from the slide. So after that, we can study this under the microscope, fluorescent microscope. And again, the positive result is a fluorescence. Okay, so like a glowing in the dark light. Okay, the second procedure for indirect FAT is one which detects antibody. Right, so in this test, a known antigen is placed on a slide and the specimen is added, which contains the antibody. So we just reverse the procedure. First, the antigen, in the first procedure, the antigen is contained in the sample. This one, the antibody is found in the serum. The preparation is then washed and fluorescent labeled anti-human globulin is added. So we expect to see, again, there will be washing in between, and we expect to see fluorescence if positive, and negative, no fluorescence. Okay, so ELISA. I want you to be very familiar with this procedure because this is a common procedure, enzyme-linked immunosorbent assay. Initially, this was used to screen for HIV infection, but nowadays there are many applications of ELISA, especially for the identification of microbial infection. Just like immunofluorescence assays, they are specific, sensitive, right? So specific, that means they can detect specifically the organism, and sensitive, even if the organism is in very small amount, we can still detect its presence. Okay, and it requires only a small amount of specimen. So that's saying the advantages offered by this procedure. Another one is the reagents are stable and have a long shelf life. So this is a very ideal type of procedure in laboratories. The results can either be qualitative or quantitative. If you do a qualitative testing, you simply have to read it directly with your eyes. But if you're going to do a quantitative testing, you will need a reader to detect for the change in color. Right, so this can also be utilized in epidemiologic surveys for automations. There are two ways to perform ELISA. One is double antibody technique, which is used to detect antigen. We also call this the sandwich assay. And then letter B is indirect, which is used to detect the antibody. Right, so let's start off with double antibody technique. Right, specific antibody is coated on the surface of the microtitration plate or a test tube. Right, so this is the equipment that we need. We need a microtitration plate or a test tube. And in those glassware or plasticware, there is a specific antibody that is already coating the surface. Okay, next we add a specimen. So the specimen can be serum or plasma, and then we incubate. We allow them to interact with each other, so there will be an antigen-antibody reaction. The next step is washing. At this point, let me tell you that if there is a need for washing, we call this method um, heterogeneous assay. But if there is no need for washing, I mean it's detected directly, it's called homogeneous assay. Enzymes are used as label in ELISA, that's why it's called enzyme linked, right? So we add this enzyme coated label antibody to the previous antigen and antibody complex. Okay, and then so you can see more or less why we call this sandwich assay. Later we have a diagram which will allow us to realize why it's called so. So after a period of incubation, okay, again we wash again, and then a substrate is added. Remember, in any enzymatic assays, there is a need to have a substrate. So the substrate, addition of substrate will bring about a change in the color of the end of the mixture. Okay, and this can be stopped by altering the pH of the reaction. Right, so the intensity of the color can be measured or can be directly measured, or I mean identified in using the eyes, naked eyes. Application. So this is very much utilized in the detection of rotavirus in young children. Since you have finished virology, I would want you to identify this rotavirus. What kind of infection is caused by this rotavirus? Right. So this is the procedure. Right. So in diagrammatic form. So first we have a microtitration plate or test tube containing the antibody, coating the surface of the antibody. After adding the specimen, which may contain the antigen, we expect that the antigen will bind to the surface of to the binding site of the antibody. Right, so we wash after, and when we add a second antibody, the second antibody, which is labeled with an enzyme. So you can see here what we are referring to as a sandwich. So there is an antibody, then antigen, then another antibody. The second step after incubation, I mean, not second, but the step after would be to wash again to remove any excess antibody and add the substrate. So after that, we can measure the color using a reader, ELISA reader, which is actually a type of spectrophotometer. Okay, so this is just the textual representation of what we have seen in the previous slide, but this is just to remind that for washing, we have to do this for more than 4 times in order to make sure that we can remove the unbound component. So I have been talking about this bound and bound component. When we say bound component, so that means that the antigen and the antibody are attached to each other, or maybe the second antibody is already attached to the complex. So that is the bound component. When we say free component, That can be the free antigen or free antibody, which we should be removing because we don't want them to interfere with the measurement. Okay, the next procedure is called indirect ELISA. So this is when we use a known antigen to identify the antibody in the serum. So again, we mix the two components, antigen plus antibody. After that, we wash, and then we add another antibody. After adding the antibody, what will happen? We have to there will be again a reaction, and then we have to wash it again to remove the unbound component. Right, so then we add the substrate. After adding the substrate, there will be a change in color, and we have to measure. We can measure this change in color in the spectrophotometer or the ELISA reader. Right, so this is the diagrammatic form. So we can see that there is a known antigen attached to the surface of the test tube of the well. After that, we add a specific antibody to be measured, which is the specimen, hoping that the specimen contains the antibody we want to check for. All right, so there will be a reaction. Then we wash, we add another antibody. This time it's labeled with the enzyme. So there will be an attachment again with the complex. Okay, we wash again, we add the substrate, there will be a change in color, and we are going to measure the change in color using the reader. I hope you see the difference between direct and indirect ELISA. Alright, so all the while I've been talking about this micro titration plate. Right, so this one is a micro titration plate. It can actually be a smaller one, thinner than this. So it's made of plastic, having many tubes or wells in it. About 96 wells, standard wells. And this would be the one that would coat the antigen or the antibody. Alright, so this is the same procedure. We just repeating the same procedure. So make sure that the plate is coated with the antigen. So if it's indirect, the patient serum, we allow it to incubate. After incubation, we wash 4 times. Okay, and then we add the secondary antibody. After that, again, incubate, then wash, then add the substrate. After the color reaction it takes place, we can measure the reaction using the spectrophotometer or ELISA reader. Right, so again, we just emphasize that if it's qualitative, we can simply use our naked eye, and there are certain patterns that we can, aside from the color reaction, there are also certain patterns that we can look for in qualitative ELISA. If it's quantitative, so again, there is a corresponding reader, and we can also perform um, serial dilution for the measurement of the titer of the antibody being detected. The third procedure is radioimmunoassay or RIA. This is a very sensitive technique that can be used to detect antigens or antibody. This was developed by Yallow and Burson, if I'm not mistaken. They are Russian scientists, but their goal then was to determine the level of insulin and anti-insulin complex in diabetics. So more or less the utilization of RIA before was limited to the measurement of hormones. The first type of RIA or RIA is conventional RIA. So this is traditional. This is a competitive method. We will know why it's called competitive later on. It measures very low concentration of antigens or antibodies. And when we say it can measure very low concentration, that means it is sensitive. So for labels, we use radioactive isotopes like heavy hydrogen, carbon 14, sulfur 35, phosphor 30, iodine 125, iodine 131. Right, so these isotopes, they are not actually molecules, but they are more like atoms, unstable atoms that can emit radiation. When we say radiation, those are the alpha, beta, gamma particles. So it's highly sensitive. We already measured that. And this can be utilized for the measurement or detection of hormones, drugs, enzymes, microbial antigens like hepatitis B antigen, CEA, alpha fetoprotein. So there are many, many applications of RIA. So it's used for the detection of antibody. And okay, RIA has three components. Right, so the components are the patient's antigen, the labeled antigen, and an antibody. This antibody is specific to both the labeled antigen and the patient's antigen. So in short, you can see more or less that, well, what I want to say rather is that the amount of antibody that is present is limited. So this antibody will be competed for by these two antigens because they are both specific to this antibody. So in short, they will be like looking for a partner, but the number of antibodies is less than the label antigen and the patient's antigen, so there will be competition. That's why it's competitive. Right, so RIA can run either as a liquid phase assay or a solid phase assay. For the solid phase assay, the antigen and the antibody are added to the mixture in a solution form, and after incubation, okay, we going to perform separation technique. So that's why if when there is separation technique, so that means there is this is a heterogeneous assay. All right, so let me tell you a little about the separation technique. Um, Separation can be done by charcoal adsorption. It can also be by um, precipitation, something like that. So by precipitation, we can remove the bound from the unbound component. Right, so next is solid phase assay, which is more common. When we say solid phase, it's just like what we did in ELISA. There is a solid structure. So the solid structure can be the test tube itself, the surface of the test tube, the surface of the um, microtitration plate, right? So it is bound with the antibody, the second antibody. Specific antibody is added in suspension. So if it is a suspension, this antibody is attached to a solid medium. For example, latex particles, polystyrene particles, right? So charcoal particles. So those are the possible media that we can utilize to make it solid phase. So in this solid phase, there is an antibody, and then we add the antigens. So what we expect to see is that there will be competition between the two antigens. After that, we're going to wash. After washing, we can do the measurement. And for the measurement, we can utilize, um, you know, scintillation counters or gamma counters in order to measure the radioactivity. Now, I have here a slide that says that your assignment in the laboratory is to look for microparticle enzyme immunoassay. At this time, RIA is no longer that popular, but it has been replaced with this procedure called microparticle enzyme immunoassay. So I want you to look for this technique. And the reason, by the way, is that RIA has many disadvantages. For example, the wastage, and of course, it's not totally safe to utilize radioisotopes in the laboratory. Right. So This is your assignment, and also IRMA or immunoradiometric assay. So number 4 procedure is Western blot. Right, so as I've told you, ELISA was the screening test used for HIV, and in order to confirm the infection, we have to use Western blot. Western blot is a test that detects viral proteins. Okay, particularly for HIV infection, that is protein P24 and P55. Right, so we can detect this as glycoprotein bands. Right, so the reason why we call it a blotting test is because we make use of a paper to blot dry the specimen. Right, so the next one is Southern blot. So it happens that this scientist, his last name is Southern. So, and then we have the Western blot. So now we have the Southern blot. But this technique is used for the detection of DNA sequences, and it can detect gene mutations, right? Like carriers for Fragile X syndrome or even sickle cell anemia. So basically, because it's for DNA sequence, this is for genetic testing. Northern blot. If we want to detect the RNA rather than the DNA, we will use the so-called Northern blot. However, this is still not well developed because it's still under research. Northern blot. We don't have an Eastern blot. Right. So let us proceed to secondary binding test. Right. So as I've told you, the primary binding tests are the one with the highest sensitivity and specificity. Now, for secondary binding tests, they are like mid-range tests. They are usually cheaper compared to the primary binding tests. Right, so they are frequently used in the laboratories because they are more simple to perform. They don't require a special equipment, no need for any readers, and they are less expensive. Usually they come in the form of kits, manufactured kits. So this is distributed to the different laboratories. Right, so agglutination is the most fundamental method or reaction in the laboratory. We make use of this technique to demonstrate interaction between antigen and antibody. So this agglutination is actually just the clumping of cells. If we make use of cells, if we make use of other materials, so again, it's just clumping together of those materials. So when antigen and antibody meets, there will be a reaction, and this will result in aggregation. Solid, something that we can see. This agglutination test can be performed in the slides or tiles. And again, this is already supplied. Sometimes this is already supplied by the manufacturers, okay, distributors, right? So, okay, we can also have tubes, perform it in test tubes. Of course, this will not be provided by the um, manufacturer, and then micro titration plates. The first one is slide or tile agglutination test. So this is rapid, easily performable per procedure techniques, and it gives a fast reaction in just a few minutes or even seconds. We can already see the result. However, this is not as sensitive as a tube method or microtitration technique. So that is the disadvantage. This can either be passive or active. We will know the difference between active and passive agglutination later on. Right, so let's start off with active agglutination. When we directly cause the agglutination of the bacterial antigen using a corresponding antibody, then it's called a active agglutination. So again, if it's direct agglutination of the antigen, it's active agglutination. Examples of this would be slide test for Salmonella, Okay, Shigella, Vibrio cholera using specific antibody also for Leptospira. Right. So agglutination test, slide agglutination test can help in identification of bacteria from cultures that are difficult to standardize and control, and also for checking for auto-agglutination. When we say auto-agglutination, the serum can cause agglutination when in the presence of cold or warm antibodies. So it automatically causes agglutination even without adding an antigen or the specimen. Next, passive agglutination slide test or tile test, because it's like made up of not glass but made up of other materials. Right. Specific antibody or non-antigen is attached to an inert particle. So this is the requirement for us to say that it is passive. Again, if it is direct agglutination of the cells or the antigens, then it's active. But if we make use of an inert particle, then it's called passive. If we make use of red cell, we call it heme agglutination. We add the word heme agglutination. Anyway, so when an antigen or antibody combines, there will be a reaction, and this can be seen as a agglutination. It can be agglutination. Right, so the particles or the carriers that we can utilize here are, as I already mentioned some of these, latex particles, carbon particles, cells of some bacteria like staphylococcal cells, right? So polystyrene, so something like that. Right, so tube agglutination is very much the same, except that instead of using the slide, of course in the slide we have to use a stick to mix the sample. Right, so for test tubes, they are more sensitive because there is a more amount of liquid is necessary to perform this test. Now for microtitration agglutination test, so this is similar to tube test, but the amount is much lesser. But even if it is small amount, it's still considered more sensitive and more economical because you have seen a microtitration plate. In one microtitration plate, you can perform like about 96 different tests. You just have to label the well using a marker. Right, next, hemagglutination inhibition antibody test or HAI. So as I mentioned, this is a passive test because we make use of red blood cells to carry the antigen. So this is used to detect viruses, especially viruses like arboviruses, influenza viruses, measles, rubella viruses. Right, so it's because these viruses can readily agglutinate the red blood cells. So that is why it's possible to do hemagglutination. Sorry, it's not passive. It is because we can just simply add directly onto the cells, and if it causes agglutination of the red cells without any binding with antigen, so that is a direct or active agglutination, hemagglutination test. Okay, but the word is inhibition. Right, so reverse positive hemagglutination. So there are other viruses that do not cause hemagglutination. So this test is for them. Right, so if the antigen is present, the red blood cells will be agglutinated. Okay, on the other hand, for HAI, so in the presence of the antigen, there will be no agglutination. That's why it's inhibition. Right, so after agglutination tests, we have precipitation tests. The requirement for agglutination test is that the antigens should be big enough, okay, or particulate. But for precipitation tests, the antigen should be soluble, right? So aside from the presence of antigen and antibody, there is a need for other things like the electrolytes that are necessary to bring about their interaction, right? So pH, temperature, so we need to provide this environment. Okay, so when they meet, instead of forming an agglutination reaction, there will be crystal lattice formation. So they come in the form of precipitates like a white color. It's very small white color particles appearing, right? They are used to detect and identify antigens in the specimen, extracts, and culture. So this procedure is for that. It's also used to detect and quantify antibodies and serum. So it's not just for qualitative testing, but also for quantitative testing by doing serial dilution. Compared with the agglutination test, it requires more experience because most of the time we miss the positive result, right? So the problem with precipitation test is sensitivity. It may not be very sensitive. Okay, how do we carry out a precipitation test? We can do it like using a tube precipitation, using gel diffusion techniques, and counter immunoelectrophoresis. The first, tube precipitation. So in tube, when we say tube, we're not talking about test tube, but rather microhematocrit tube or capillary tube. Right, solution containing the antigen, so this is the specimen, is layered on an antiserum, so this is the antibody. This is the reagent. After that, in the point of contact, after incubation, there will be formation of precipitative positive, and if negative, there will be no precipitation. So again, in the point of contact. Disadvantage. So one is it requires large amount of antiserum, so it's not really very economical, okay, and it's not very sensitive. However, it can be used for the detection of antigens found in the CSF like HIB. I'm not saying HIV, but Haemophilus influenza type B. And also for grouping of beta hemolytic streptococci. If you remember, there are many strains of streptococcus, and one of them is beta hemolytic strep, complete hemolysis. Gel diffusion test. There are several ways to perform this. It can be single diffusion or double diffusion. The requirement here is an agar gel. So we need a petri dish. We need a petri dish. We have to layer it with agar. Right, so the agar may or may not contain the antigen or the antibody. Okay, so for double diffusion, it doesn't contain the antigen or the antibody. Instead, we will be placing wells. We're going to puncture holes on the surface of the agar, and we're going to place the antiserum and the antigen. And after incubation, they will diffuse, right? Diffuse out of the gel. After diffusion, there will come a point that they will meet the antigen and the antibody. So once they meet each other in the point of contact, there will be precipitation, right? So for single diffusion, this is when we add the antibody or the antigen directly onto the gel. And then we expect by puncturing a hole in the center, and then we add maybe the specimen, there will be precipitation. So that means that the antibody corresponds to the antigen that is present in the agar. Okay, so double gel diffusion, as I have described, is also called Ochterlony method. Right, so this technique is employed in ELEC test for diphtheria, identification of toxogenic strains of diphtheria, and beacon test or beacon test used for the detection of enterotoxigenic Escherichia coli toxin producing. Now for single diffusion technique, I've already mentioned this. Ring of precipitation will form around the well, okay, and that would correspond to the antigen that is present. Okay, so next one is counter-immune electrophoresis. In this case, we apply because antigens and antibodies are both, um, shall we say, protein in nature or glycoprotein in nature. It is possible to apply electricity and they will migrate. For example, um, we have the proteins that are present, the antigens that are present, and then after adding the antibody, we're going to apply electricity, and this will force the antigen to meet the corresponding antibody, and there will be lines of identity that will be formed in the process. Right, so number 3, another still under secondary binding tests, are the complement fixation tests or CFTs. So this is a very, not really an easy test to perform, right? So it's better to perform this not in the main laboratory, I mean in the common laboratory. Right, so the method. Antigen is mixed with the serum if you want to test for antibody, right? And then we add the complement. So you know what happens when the complement is added, right? So antigen, when present, will attach to the antibody, and then we add erythrocytes that is coated with antibodies. What will happen? Okay, so since the complement is already fixed by the antigen-antibody complex, it can no longer cause the um, any lysis. However, if after adding the erythrocyte, okay, and there is lysis, so that indicates that the antigen is not present. So that is why we call it complement fixation. We fix the complement. We make the complement busy so that it will no longer be able to bind and lyse the red blood cells. Right, so this is the second to the last slide. Tertiary binding test. I have mentioned that this is a test that is most commonly done in research laboratories, and this is performed in vivo. Right. Next, there are certain factors that can affect the antigen-antibody reaction, like specificity. So sometimes if I'm going to talk about that, let's say cross-reactivity. There are some conditions that produce antigens that are closely related, and it can lead to a false positive result. Temperature, so if we do it at a very high temperature, since they are proteins, it can lead to denaturation. Again, the proper pH can also be a factor, ionic strength and concentration. So these are all related to the optimum condition for antigen-antibody reaction, and also intermolecular specificity. And at this point, I would like to mention of the so-called the pro-zone and the post-zone phenomenon. So I want you to look into that also. Okay, zonal reaction effects. Thank you.